Progressive Society of Education, Modern College of Pharmacy, Nigdi, present a topic of Fourier Transform Infrared Spectrometer. This topic is presented by Vedasri Dhumkekar, Devendra Dudhade, Girish Punde, Anish Gaikwad, and Shubham Galande. Introduction. An infrared spectrum represents a fingerprint of sample with absorption peaks which correspond to the frequencies of vibration between the bonds of the atoms making up the material because each different material is unique combination of atoms. No two compounds produce a exact same spectrum, therefore IR can result in a unique identification of every different kind of material. FTIR is a powerful tool for the identification the type of chemical bond present in the molecule. FTIR have the greatly extended the capabilities of IR and applied to many areas that are difficult to analyze by the dispersive instrument. In the dispersive IR light is separated into its individual frequency by the dispersion using a monochromatic grating. All the frequencies and intensities can be simultaneously determined. FTIR gives a plot of intensity versus frequency. The instrument used for the FTIR is scanning Michelson interferometer. FTIR frequencies are allowed to the intersect to produce and the interference pattern, and this pattern is analyzed mathematically using Fourier transform to determine the individual frequencies and their intensities. FTIR instrumentation. The Michelson interferometer consists of following parts. First, a moving mirror. Second, a fixed mirror. Third, a beam splitter. Fourth, a radiation source. And fifth is FTIR detector. Let's introduce a sample cells. To obtain an spectrum, the sample must be placed in the container or the cell that is transparent in the IR region of the spectrum. Sodium chloride or the salt pellets are common means of placing the sample in the light beam of the instrument. Let's talk about sources. First one is a nurse glower, hollow rod about 2 mm in diameter and 30 mm in length, composed of the rare earth oxide. Externally heated. Their temperature is about 1000 to 1800 degrees Celsius. Radiation over the wide wavelength range. Second one is a glower source. The rod of the centered silicon carbide 50 mm in the length and 4 to 8 mm in diameter, self-starting. This is heated about the temperature between the 1300 to 1700 degrees Celsius. Tungsten incandescent lamp, black body source used in near IR instrument. Second one is a nichrome rhodium wire, coiled, heated, emits in the mid IR. Black oxide layer is formation in the nichrome or rhodium wire. FTIR detectors. There are the two types of the detector. First one is a thermal detector and second one is a photon detector. In the thermal detectors, their first one is thermocouple detector. The two junctions of the dissimilar metals connected together. Electricity is directly proportional to the energy difference between two connections. The second one is a bolometer. One arm of the Wheatstone bridge and similar strip of metal is used as balancing arm of the bridge which is not exposed to the radiation. Amount of the current flowing is directly proportional to the amount of radiation on the detector. Second one is a photon detector. Interaction between the incident photon and the semiconductor. Energy from photon is tricking electron in the detector and raises them from the non-conducting to the conductance state. Example is a MCT detector, pyroelectric detectors, DTGS detector. Interferogram. The interferogram consists of two modulated waves. First one is a constructive interference and second one is a destructive interference. Advantages. Flaget advantages, high speed. Jacquard advantages, high sensitivity. Slitless system. All of energy of the source is utilized in FTIR. Rapid scan time with high sensitivity. Cone's advantage. Disadvantages. The first one is a FTIR instruments have a single beam, whereas dispersive instruments usually have a double beam. The second one is interferograms are difficult to interpret without first performing a Fourier transform to produce a spectrum.